Okay, that sounds great. Thanks for letting us know.
Well, good afternoon. I think it's right 1 p.m. in the afternoon. We can get started. This is room number 1 of poster presentations by junior investigators, and we have 4 presenters today. We're going to have 5 minutes for each presenter, followed by a couple questions for 2 minutes or so. And then at the end of this session, we're going to have a nice discussion to think about collaboration possibilities and talk about other things that are related to our research interests. I'm going to start the timer, and I'm going to let you know when we hit 4 minutes, 30 seconds, so you have an idea how to wrap things up. So without further ado, I would like to start with Heather Baumberger, and she's from Biomedical Engineering Department, working in David Wood's lab. And she's going to give us the poster presentation on in vitro investigation of natural killer cancer cell interactions and metastases. Heather, please take it away. You may want to unmute yourself.
There we go. Yep. Hi, everyone. My name is Heather, and I'm going to talk about my research on NK, or natural killer, and tumor cell interactions in metastasis. So first, NK cells are very well suited for cancer immunotherapy, partly because of their role in tumor surveillance and clearance, which is why we're so interested in them. They're also well suited because they don't require any priming or any pre-stimulation. They just act through a balance of activating and inhibiting receptors on their surface and in the environment, and they sometimes can act through something known as antibody-dependent cellular cytotoxicity, or ADCC. And this ADCC function can be exploited by drugs, and in this case, I'm going to talk about a drug called a TRIKE, or a tri-specific killer engager. This one specifically is made by our collaborators in Martine Felici's labs, and Martine's lab, and it has 3 arms. The first one is CD16, which is a NK activating surface receptor, and B7H3, which is known to be upregulated on the surface of cancer cells, and IL-15, which is, of course, an NK activating cytokine. So this serves as a synthetic antibody in this ADCC process, and once it binds these 2 cell types, it sort of forms a killing synapse between the 2. And once that's formed, the NK cell can release granzymes and perforin to induce apoptosis in the tumor cell and cytokines to further the immune response. So one reason we want to study this TRIKE specifically in a microfluidic model is because mouse NK cells do not have this ADCC function capability. And so we really want a physiologically relevant humanized model to be able to study what this looks like in humans as a drug. And so some preliminary data on this drug in vitro on a 2D surface. So we did a functional assay with 2 target cells that we would be using in our microfluidic model that I'll talk about in a second. First, HUVEC, which is our endothelial cell model, and MDA-MB231s, which is our breast cancer cell line that we're using. So we put NK cells and one of these 2 cell lines on a low adhesion surface. And with no treatment, one of the individual components of the TRIKE or the entire TRIKE construct, and then we can calculate % killing. So there's really no killing at all for the HUVECs, and there's significantly more killing for the 231s with the entire TRIKE construct. So in addition to studying the role of this TRIKE in a physiologic humanized model, we also want to study this role of tumor surveillance and clearance in general. So we want to better understand how NK cells can do this and how they can migrate and function in metastasis. So our biggest question here is, where exactly do NK cells attack? once we can understand where they attack in the metastatic process, we can understand that environment better, and then hopefully we can understand how to therapeutically manipulate that environment. So do NK cells attack when cancer cells are in circulation in the blood vessel? Do they attack slightly later in the process after cancer cells have adhered or maybe become stuck in the vessel? Or is it even later after they've extravasated into the tissue? So for the sake of time, I'm just going to go over these two stages here. And I have one microfluidic model that I can set up differently to model the slightly earlier stage of adhesion and the slightly later stage of extravasation. So here's that model. Here's a macroscopic and a microscopic image. As you can see, it's 2 channels, and I'll just quickly go through how we set that up. First, in this lower channel, we can perfuse collagen, allow that to polymerize. In the upper channel, we can perfuse HUVEC and allow this to become confluent and form a lumen. And then we flow through fluorescently labeled MDA MB231s. And so here's the point where we set this up slightly differently. So we either allow the cancer cells to adhere for 3 hours and then add our primary human NK cells for the adhesion model, or we give the 231s a full 48 hours to adhere and extravasate. So for the extravasation model. So we can either, at the point we introduce the NK cells, we either have a model of the slightly earlier or the slightly later phase of metastasis. And at that point, we can just image and quantify. So here's just a quick image of what that looks like. In the model, MDA and B231s are red, pink are the NK, and green is a caspase 37, which is an apoptosis marker, and one image of an NK cell interacting with a cancer cell and inducing apoptosis. Thanks very much. Are there any questions?
Fantastic. Thank you so much. And sorry for rushing you through, but
No, it's okay.
you know, David is looking over my shoulder, so he would give me bad points if I didn't. Are there any questions, quick questions from the audience? If you do, please unmute yourselves. And
so I just missed something. Maybe you talked that. Did you add the trike drug there, or are you just testing a concept to see if the model works?
Yeah, good question. So at this point, the trike has just been tested in 2D, and we are optimizing the model to be able to eventually study the trike and potentially other drugs in our model. So I'm actually working on that right now. I was in lab super late last night working on that exact experiment. So hopefully I'll have that data very, very soon.
And I understand that you actually use the HUVEC cell line as a negative control, right?
Yeah, so exactly in that functional assay, right? So we just want to make sure that in our model, the NK cells only kill the cancer cells, which is, so the cancer cells would express that B7H, that level of B7H3, and then the trike can sort of target the.
So 231s are pretty aggressive breast cancer cell line. Is there a reason why you picked that?
So that's just kind of that's a model that we've used pretty regularly in our lab, in our microfluidic models. It does well in the microfluidic models. We also have that model, or that cell line, fluorescently labeled. And in the past, we've also worked with collaborators that were interested specifically in breast cancer, or triple negative cancer, breast cancer even more specifically. So that's kind of just why. And we kind of have an idea of how they behave in our models. So that's kind of why we're using them.
Fantastic, thank you. Any other questions?
So that's still kind of a 2D model, right?
So it's well, now I don't have it sure, but so the layer of endothelial cells is a monolayer, but then the collagen is about 100 microns tall, and a similar, you know, it's 3D. The collagen gel is a 3D gel within that channel. So it's got, you know, space in the X, Y, and Z planes. And when the NK cells and cancer cells invade, they are invading that collagen gel, if that makes sense. So there are in the vessel, the endothelial cells actually line the you know, all channels of the, or all walls of that upper channel. So I have I stopped sharing my slide, but I had an extra
That's okay. Yeah,
spare.
I don't take much time to.
Yeah, I had a spare slide basically of a slice of that upper channel with CD31 staining that shows that it is all the way around the channel. But yeah, thank you. That's a good question.
Good questions. Yeah, we can continue discussing it in the second part of this session probably. And thank you for the presentation, Heather. And the second, we have actually Elizabeth Christ from David Wood's lab for a change from Biomedical Engineering Department. And she's going to be talking about recapitulating the metastatic cascade in vitro model to study by multimodal breast cancer cell migration. Go ahead, Elizabeth.
Thank you. Yeah, so my name is Lizzie, and my project focuses on studying multimodal breast cancer cell migration throughout the metastatic cascade, and I aim to do this by developing a metastasis mimetic model that recapitulates the stages of metastasis in sequence. So the reason we want to study metastasis is because this process is the cause of over 90% of cancer-related patient deaths. And in breast cancer, survival rates actually drop from 90% to 27% once those cancer cells have metastasized to distant tissues. The cascade can be broadly described by 5 steps, beginning with local invasion at the primary site, intravasation or entry into blood vessels or lymphatic systems, circulation, exit out of the blood vessels or lymphatic system called extravasation, and finally colonization of a new tissue. Cancer cell migration as either single cells or collective units is integral to metastatic progression, and it's known to be highly plastic as both single cell and collective migration, as well as transitions between the two forms, are known to be contributors to breast cancer metastasis. However, we don't know which form or forms are most prevalent at individual stages of metastasis. My central hypothesis is that collectively migrating units of breast cancer cells will have overall greater metastatic potential because they've been shown to have around 23 to 50 times the metastatic potential in murine models of breast cancer. Because they're able to evade immune cells, they're able to avoid apoptosis and seed secondary metastases. So to date, though, the contributions of multimodal cancer cell migration to metastasis, in particular collective migration, has been poorly understood, as the majority of in vitro bioengineered cancer models have predominantly relied on modeling tumors using single cancer cell suspensions. They also tend to be highly reductionist in that they only recapitulate a subset of the events in metastasis. In vivo models, on the other hand, are hindered by their biological complexity, they're costly and labor intensive. And so there's a need to develop an in vitro model of metastasis that more accurately mimics in vivo pathophysiologic conditions of tumors, integrates each stage of metastasis, and enables visual monitoring of single cell and collective migration through a simplified, cost-effective, and optically accessible design. And so to begin creating this new model metastasis, I began some preliminary work into optimizing protocols to generate multicellular tumor spheroids to mimic the primary tumors. And so I'm using 2 cell lines that are metastatic and mesenchymal 231s and HS578Ts, as well as a non-tumorigenic normal breast epithelial cell line MCF10As. The spheroids composed of monoculture 231s and HS578Ts are going to be used to study single cell migration since they are mesenchymal, whereas co-cultures of the mesenchymal cell line and the normal 10As will be used to study collective migration, since this has been shown to initiate collective migration in past work. And so we see in these representative images of the spheroids, as well as quantification of their diameter, that we're able to consistently generate spheroids of greater than 500 microns in diameter after 5 days of growth, which is important for the development of oxygen and nutrient gradients that mimic in vivo tumors. And so to demonstrate that multimodal migration is achieved using this system, we embed these spheroids into 3D collagen hydrogels and track their migration over time. In the monoculture condition, we observed high occurrences of single cell migration over time. as well as rapid spheroid expansion due to both proliferation and migration over 6 days. Interestingly, though, we did also observe some less frequent, consistent, coordinated multicellular migration in the form of strands, as you can see here. In the co-culture conditions, we found some unanticipated results in that the 231s plus the 10As did not really generate any sort of collective migration, and we believe this is because the 231s proliferated so quickly that they clogged the migration of the 10As. The HS57AT co-culture with 10As, though, did lead to some collective migration, and as a control, the 10A monoculture condition did not really generate any migration as well, which would be expected. And so while we are excited about this, that we will be able to study multimodal migration using this more physiologically relevant model of a primary tumor, we do want to know how these forms of migration propagate throughout metastasis. And so I developed this metastasis mimetic model where you can see in the primary site, it resembles a modified Transwell assay where there's a bottom layer that is a fluidic channel that connects both the primary site and secondary site that is coated with endothelial cells to represent a blood vessel. And then there's a upper level, top level tumor chamber where I am able to embed a multicellular tumor spheroid in collagen. And those two compartments are separated by a thin porous membrane. And so you can see that I'm able to add an individual spheroid embedded in collagen into that primary site and actually track the growth and migration of that tumor over time. And then that secondary site is actually developed as an extravasation model from our lab, and we have been previously shown how we can quantify extravasation and metastatic potential. And so using this metastasis mimetic model, we were able to quantify for the first time the propagation of multimodal cancer cell migration throughout the course of metastasis that is initiated from a physiologically relevant model of a primary breast tumor. So that is all right now, but there's a lot of exciting new steps, so I'm happy to collaborate. And I'll take any questions. Thank you.
Thank you so much. That was a great presentation. And thank you for stopping right on time. Any questions? Okay, hydrogels. What kind of a hydrogel did you use for that?
We use collagen right now, collagen type 1. And I've been using it at a couple different densities, but I actually plan to use some decellularized extracellular matrix from actual organs eventually, especially in that secondary site to mimic some of that organotrophic signaling during metastasis.
So is it only collagen, or is it kind of mixture of basal membrane proteins with the main ingredient being collagen?
I only add collagen, but I guess I can presume that the cells themselves are probably laying down some of their own extracellular matrix like fibronectin, collagen 4, laminin. I have not stained for that, but that's definitely something I could carry out.
Okay. Any other questions?
Elizabeth, hi, this is Brandon Burbeck.
Hi.
Sorry, I'm trying to get my camera to turn on. How are you guys? In those experiments, wait, did you have a co-culture with MCFs 10 and 231? Is that how that was set up? I used to do this kind of thing a long time ago. I'm wondering if, like the MCF10A are so kind of clustered together because of like their cadherin junctions and things that they just can't migrate. I mean, is that kind of embedded in your question? Maybe I didn't understand how it fits in kind of at a big picture.
Yeah, they definitely. So what's interesting about the 10As is that they're very confluence dependent in terms of their expression of E-cadherin or N-cadherin.
Yeah.
And so if they're not confluent, they will start to become more mesenchymal. But of course, when I make a spheroid, yes, they are very compact in that core. But what happened was when they were cultured with the 231s, I think the 231s proliferated so much that it created like a back pressure that basically inhibited those 10As from migrating. But when I cultured them with the 578 T cells that are still mesenchymal, they don't proliferate as much, which allowed those 10As then to basically participate in like leader follower migration, where there'd be like an HS578T green cell followed by like a red cell.
Okay. So then kind of the bigger question, maybe I just didn't grasp that, not because of you, just because I wasn't trying hard enough, but like you're kind of wondering if the the worst cancer cells are kind of encouraging the transition of the kind of better, more normal cells to take on a bad phenotype. Is that?
Yeah, exactly.
Have you tried?
Yeah, so
and you're preforming the spheroids. Like, what happens if you, like when I did this kind of thing, I would just seed cells at more the single level and then let them form their own spheroids into matrigel or whatever, collagen 1. Does that change the dynamic if you let them form their own spheroids and then induce migration? Or
yeah, so I do grow the spheroids for 5 days just in like a low adhesion 96 well plate, and I like put a single cell suspension, and then over those 5 days they compact because of that low adhesion environment. And then I take that naturally formed spheroid and place it into collagen, which then allows for that 3D migration, because when the spheroid is growing, it's suspended in media, so there's not really an ability to migrate. There's nothing to grab onto.
Yeah. Okay.
Thank you so much for the
Interesting.
questions. I think we're gonna continue with our next presenter, Amanda Salzwedel from Provenzano Lab at Department of Biomedical Engineering, and she's going to be talking to us about the impact of irreversible electroporation combinations with immunotherapy in stimulating anti-tumor immunity and eliminating PDA-resistant barriers. Amanda?
Can anybody see? Okay, great. Thank you very much for the introduction. Pancreatic cancer is the fourth leading cause of cancer-related death in the United States. The expected survival is about 3 to 4 months post-diagnosis. The 5-year survival is only between 8 and 9%, and right now there are not really highly effective treatments to treat the disease, to manage the disease. So the aspect of the tumor is this very hard nodular tumor like that. But if you get a closer look at that, there's a lot of complexity to these tumors that could play a role in this therapeutic failure during treatment. So one of the main components here that they play an important role on that is the tumor stroma. So the tumor stroma of pancreatic cancer is basically composed of hyaluronic acid, proteoglycan, and collagen. And this combination of hyaluronic acid and proteoglycan is highly hydrophilic in forming this gel that attracts a lot of water to it, that expands about more than 40 times its original size. But because pancreatic cancer is highly desmoplastic and has this high, a lot of collagen laid down to it, the collagen kind of restricts the expansion of this gel, and that causes a very high increase in intratumoral pressure that leads to a lot of the therapeutic challenges during treatment of pancreatic cancer. Like, for example, this high pressure inside the tumor causes the collapse of blood vessels, which greatly diminish drug delivery during treatment. And it's not only the collapse of these vessels, the nature of pancreatic cancer is these high desmoplastic tumors, basically because of the presence of cancer-associated fibroblasts that lay down a lot of this very dense collagen matrix that are going to inhibit infiltration of immune cells and also are going to contribute to this very high pressure inside the tumors. And pancreatic cancer is a highly metastatic disease, and in order to have a good outcome out of these treatments, you have to have the formation of anti-tumor immune response. But unfortunately, the tumor microenvironment of pancreatic cancer is highly immune suppressed, and that greatly contributes to the poor survival to the disease. So in order to overcome these challenges and develop therapies that can eliminate these barriers, our lab focused in, for example, the use of irreversible electroporation methods combined with immunotherapy. So irreversible electroporation is a surgical ablation technique, which is basically the introduction of these 2 electrode needles here, and they generate an electric field, which is converted to a thermic field that's going to cause cell death in that treated area. So it has been reported in the literature that improves the drug delivery in opening of vessels, that the cells die by necrosis, and you stimulate this focal inflammation that kind of causes this immunological awakening at the treatment site, which is an optimal concept for immunosuppressed tumor microenvironments such as the pancreatic cancer. So the goal of my project right now was to compare the performance of two needles when treating pancreatic cancer, this single needle IRE and this double needle IRE. And with that, after defining the needle, I was planning to combine that with the immune checkpoint blockades and IL-2. So we have this model in our lab, which is the KPC model, that represents with fidelity all the therapeutic challenges of pancreatic cancer in humans. So what we did is that we treat these tumors with both of these needles, and we saw that these needles were successfully able to induce necrosis in the tumors, no matter the type of needle that was used. And not only that, there is a much higher level of necrosis on the treated tumors, and a decrease in proliferation Ki-67, those tumors as well, and also immunological stimulation, some sort of immunological awakening as well, as there is also more migration of CD45 positive cells to these IRE treated sites. Not only that, we saw that around these necrotic areas, there was a higher accumulation of M1 macrophages that are iNOS positive, and also a co-localization of TCD8 cells, which is correlated with formation of potential anti-tumor immune response or immunological awakening. So we also saw that the IRE in these models improved drug delivery in the IRE treated areas. As you can see here by the red, the doxorubicin was able to penetrate the tumors better. And we saw that that happened mostly because there was a higher opening of vessels in the IRE treated areas compared to the sham areas. But because the pancreatic cancer is a highly metastatic disease, now that we developed all these concepts and saw that the needle work and achieve the goals that are reported in the literature in our KPC model, we developed this metastatic lung metastasis model so we can see if you can develop an effective immunotherapy protocol. And what we are doing right now is like getting these animals and treating them with a combination of IRE, immunocheckpoint blockade, and IL-2. And because pancreatic cancer is a polyimmunogenic tumor, we are planning to vaccinate the animals or give a second boost of tumor lysates to these animals to emphasize and reinforce the potential immune response that these animals could develop. And then we can analyze the immune response and survival of the animals. And with that, I would like to thank you, everybody, for attending this presentation, and especially my mentor, Dr. Paulo Provenzano. And if you have any questions, I would be happy to answer. Thank you.
Thank you so much, Amanda. Fantastic presentation, a lot of data. I think I feel bad to push you to present everything in 5 minutes. Any questions to Amanda?
Amanda, do you know if that invasion of the macrophages was a result of having the needles injected into the mouse, or were you able to separate that out? Like, were the needles just inducing an immune reaction?
We actually, there are some regions when you treat with a double needle IRE, there are very obvious lesion areas. Is that your question? And that's the areas that we focus on for our analysis. And also there was when you get the tumor slices, you kind of mark in the slide where these areas are for the IHC, and then we make the cuts in those areas as well. So, and we, for example, for the double needle IRE, we have a single point of treatment that was very obvious because of it causes very intense tumor debulking. But for the single needle IRE, I could not really correlate it like as well as the double needle because I had multiple points of treatment. So it's like several little points in the surface of the tumor. But in comparison with the sham and the amount of necrosis between the treated areas and the sham tumors, we have a pretty good guess that the necrotic areas in the IRE treated areas are corresponding to the areas of treatment.
Nice. I was going to ask a similar question, but I think probably I'm going to wait just to stimulate some more discussions until later. It's a very nice presentation. Let's continue. And our final speaker of this session is Huda Zahid, and she's coming us from chemistry department, development of small molecule inhibitors of the epigenetic protein EPTF. Huda, please go ahead.
Okay, so I'm Huda. I'm a fourth-year graduate student in the Department of Chemistry. My presentation is going to be pretty different from everything else we've heard today. I'm working on the development of chemical inhibitors for an epigenetic protein called EPTF. Just a little introduction. So all of the cells or most of the cells in our body have essentially the same DNA sequence, but your liver cells are very different from, let's say, your heart cells or your muscle cells. And that's governed by epigenetics. And that's at what point or which cells express or turn which genes on or off. And so DNA in our body is wrapped around these proteins called histones, shown in red. And when these are really compact, close together, they're not accessible by transcription machinery. So those genes are turned off. However, these histone tails can get modified. For example, they can get acetylated, and the presence of this acetyl group will turn these genes on by allowing the chromatin to sort of become loose and more accessible by transcription machinery. One of those proteins that interact with these acetyl-lysine groups are called bromodomains. And once they interact with the groups, they will be able to recruit more transcription factors towards this specific site and turn the genes on or off. So my work focuses on one of those proteins. There are 61 of those in that family, and BPTF is an understudied protein involved in a number of cancers, such as melanoma, lung cancer, and leukemia. It's a multi-domain protein, which means that the bromodomain is a part of a larger structure, and which is why there is a need for potent and selective inhibitors that target that specific domain and do not target the rest of the domains or rest of the proteins in that family. So when I started working on this project, we started with this scaffold that was sort of moderate affinity for BPTF. And after many iterations of organic synthesis, I was able to serendipitously come across this molecule here, which is actually a 70 nanomolar inhibitor of the protein. And this is probably the most potent molecule for this protein so far reported in the literature. Using these set of molecules, I was able to design and optimize a few biophysical in vitro assays based on NMR, as well as a bead-based assay AlphaScreen. And we were able to obtain a bunch of extra crystal structures, which were the first ones for this protein and molecule. Moving on from there, my future directions are to use these molecules in cancer models, starting with cancer cells such as K562, as well as lung cancers, where these proteins, where BPTF is overexpressed, and see whether my inhibitor can actually selectively target that protein. Going into a little bit detail about the assays I helped optimize in our lab. This is a bead-based assay where I use donor and acceptor beads, where my protein is attached to the acceptor beads. Then I have another binding partner. And when they're close together and you excite one of the beads, singlet oxygen flows, and you can measure the signal. Then you add in your inhibitor in increasing concentrations, and you can get a dose-dependent curve and actually measure the IC50s. And this is actually one of the first assays in our lab that can actually work, that actually works for this protein. So this really pushed the project forward, and we were then able to also obtain, using some of our molecules, one of the first co-crystal structures, and now we can actually use extra crystal guided hypotheses to design more selective and potent inhibitors. So this is all a very traditional medicinal chemistry project, but for my future work in collaboration with the IEM Fellowship, I'm going to work in collaboration with the ITDD, starting with cheminformatics with a large library of molecules, virtually screen them using our protein target extra structures that I have at the hit compounds, and then we already have the tools set up to validate those hits and identify them in vitro and then eventually in vivo. And that is all for today.
Thank you so much. Great presentation. I have a bunch of questions, but before I ask my questions, are there any questions from the audience?
I see in the panel C of that, you have the MCF7 breast cancer cell lines. So do other breast cancer cell lines express this, and could they be potential targets, like triple negative, like the 231s and 578Ts that Heather and I use?
Yeah, that's a good question. So to be honest, our lab doesn't work on cell culture stuff right now. We're just starting to get into this. So this initial data is with a collaborator, and they tested a bunch of cell lines and eventually concluded that with MCF7 cells, they're actually sensitive to both kinds of molecules, although one of them targets the protein and the other is a negative control. And then K562 cell lines in red is the positive control that actually targets that cell line. So they were able to sort of identify which cell line is sensitive to that specific effect. But again, we are just starting to get into this, and yeah, it's very, very preliminary stuff.
If I may, so I think I read in your abstract that the BPTF, not all the function of BPTF is known practically. So there was some question about fully inhibiting it and not knowing exactly what kind of effects or other functions it plays in the body. So how do you walk that fine line between completely inactivating it versus causing effects?
Yeah, that's a good question. So one of the things I talked about in the abstract was that BPTF is a really big protein, like 3,000 amino acids. And the bromodomain that we're trying to target is just one domain with at least 4 other known domains that have distinct functions. And this is where my molecules come in, because if they're targeting all 5 or 6 of the domains, then you can't really tease apart their actual physiological function, or whatever phenotype you end up seeing might be the result of some off-target effects. So once I have like a potent molecule, I also screen them for selectivity. So I would measure their affinities with each of the domains or with other proteins and make sure they're at least 70 or 80-fold selective, which is the sort of the gold standard for chemical probes. And that's how you sort of move towards selective inhibitors.
Cool. Any other burning questions? Okay, so then we can slowly transition in part 2. And in part 2, our purpose is to look at opportunities to collaborate within this group or within the outside, and understand what collaboration maybe means, just to discuss it, and how you think your project may benefit from collaborations. And the main purpose of that, or one of the main purposes of that, is to be able to target this pilot project competition that's going to be for junior investigators like yourselves. And it's going to be funding from IEM in the order of $5,000. And to be able to practically pursue some interesting ideas that you may not be able to do potentially by yourselves. Is anybody interested in starting a conversation about what you would, for example, expect from a collaboration? How would you define a good collaboration? And what are the things that you think in your project specifically or more broadly that can actually benefit from somebody else, maybe from a completely different field or from a same field?
I mean, I think in general, like what makes a good collaboration, probably somewhat equal contribution, whether you're bringing expertise because you have a model and then someone else has like more of a biology question, and then you can combine those. So two ideas that kind of came into my mind, and I'd love to hear feedback, but Amanda, I know that you're studying PDAC, and so it's like a hypovascularized tumor, whereas like I'm looking at breast cancer, which would be normal vascularized. So I'm trying to think about like if I were to use my metastasis model that does have like a blood vessel, like would that blood vessel essentially just not be like needed? Or because the tumor itself doesn't actually have vascularization around it, would that actually be a decently useful model to study metastasis from a pancreatic carcinoma?
I think your model is actually extremely useful. I think that maybe the pressure in the cells, you could put a hypoxic chamber in your model. You kind of simulate the hypoxia in the tumor, right? I think your model is pretty. Like,
yeah, well, the tumor itself is large enough where the core is already hypoxic and necrotic. Just because like the size of the tumor is already big enough where the diffusion of oxygen and nutrients to the center is already limited.
I think I missed the point of your question. I'm sorry. You're talking about pancreatic cancer or breast cancer?
Pancreatic cancer, like could I take a cell line of pancreatic cancer cells, make spheroids, and then try to simulate metastasis and answer a
Yeah.
question that you've been wondering
I
about?
think so. I think that that's actually a very valid model that you have. And this hypoxia point that you brought is extremely important, because the cells that contribute to metastasis could be cells that have morphological changes, right? That they are more metastatic, but also the cancer stem cells are a great progenitor of the metastasis as well. And they do behave extremely differently in a hypoxic environment, right? So I think that I think that your model is perfect, but I don't know how you culture the cells. So what's the condition that you culture the cells? So I think that there are several ways to put that in a more anaerobic condition, and you would reflect the phenotypes of the cells in pancreatic cancer better. And if you want even to test the drug response to your formation of metastasis or so, I think that representing the anaerobic condition of pancreatic cancer is crucial for you to have an accurate response, because I am very aware that there's like upregulation of pumps in cells, right? That they can like exclude like chemotherapy drugs in like different conditions. So I think that you can perfectly use your model, just like cultivating it in a more like anaerobic type of environment. You know, but still the surface of pancreatic cancer is fairly oxygenated by the blood vessels, right? So as long as you can grow, I think that even if you can grow a little tumor that with a big enough to have a hypoxic center, maybe you don't even need to change the conditions of culture. But
that's what I just make that assumption in what I've been doing with breast cancer. So like in theory, I could grow up pancreatic cancer cells, make the spheroids or the tumors, make sure that they're large enough to naturally develop that hypoxic core, and then just place it in the model and leave it oxygenated, because we know that the core is not.
But I think more important than that is the representation of the matrix.
Okay.
You know, so the representation of the matrix, like the collagen and the cancer-associated fibroblasts, these are the main therapeutic, barriers for treating pancreatic cancer. And I was even thinking, can you just put a human tumor there, a piece of human tumor in this type of model? And just,
that's what I would eventually want to do.
Yeah, that would be even better if you can have a PDAC piece that represents fully the tumor. And even you can even use, because the natural killer too, in my PhD project, at least the natural killer was highly involved when you use cytokines to treat, which I'm using cytokines right now too. Maybe, oh, I kind of lost my train of thought here. Then the trike drug could be used, because I'm kind of a pharmacology background, so I only think about drug. So you can use your model to check migration, metastasis, and stuff like that, and with the piece of human tumor, and then we can actually test the trike drug, right, with the human. Can you do something like that too, or no?
Yeah.
With the cells, the human natural killer cells.
Yeah, so that'd be like a three-way collaboration with like Heather.
Yeah.
And getting her natural killer cells in there. Because in theory, you could feed in the natural killer cells in the blood vessel and have them go attack potentially the tumor. Or if the natural killer cells just stay in the blood vessel, any cancer cells that intravasate and end up in the circulation could in theory be killed off by circulating natural killer cells.
Yeah, I was wondering even like if you, not even like that, but can you just like treat an animal with IRE, for example, and then excise that tumor slice fairly fresh, and then put it there and track how it's going to be like invasion and stuff like that, you know,
and see if it's changed.
And see if it changes at least the metastatic potential of the tumors, you know, like with IRE or even combination of other drugs as well. I think your model is perfect to check combination, right? Combination therapy.
Yeah, that's a good point.
So, yeah.
How about the chemistry angle?
Well, I mean, if you're drug is able to be used or tested on other breast cancer cell lines. Like we have plenty in our lab to do a quick test. And then, you know, again, using like my metastasis model, we could see if that drug inhibits the metastatic potential of the cancer cells.
And we can add any cancer cell line. So if there was one in particular you were interested in with your epigenetic marker, like we could add that cell line, we could add the drug.
I think those metastatic models might be pretty helpful because so far we've only tested our compounds in just like regular cell lines, not any kind of organoid models or anything. So that would be definitely helpful.
So you're currently testing on like just like in 2D, like cells are plated and you add it in.
Yeah.
Yeah, I would imagine that there's probably pretty big epigenetic differences between a 2D model and a 3D model. So that would probably be pretty, you know, important to check. And I assume you probably do a lot of in vivo experiments, or no?
Not a lot. So we just have collaborators, and to be honest, we didn't have a good enough molecule. So the series that we just stumbled upon right now, we're just starting to get into the in vivo
Gotcha, yeah.
work. So it's just really in the works right now.
Yeah, yeah. So that's kind of of course, Lizzie had a nice intro on how like microfluidics are a good sort of in between in vivo and traditional in vitro models. So it's helpful to see sometimes that 2D, you know, it's helpful to see more, you can see more clearly in a microfluidic model what's going on in 3D rather than 2D. So, and I'm sure that I'd be interested to see, you know, kind of more specifically the epigenetic changes that would take place in a 2D versus 3D.
That's okay. So we have 2 ideas so far, if not 3. So I'm going to continue stirring the pot a little bit, and now I'm going to ask Amanda. So the tumors that you're treating, of course, you're doing it with the animal studies, and you're inserting the RF probe in there. You're heating it. You're applying an electrical field. You're applying heating, and then you're stimulating an immune response. The tissue is complex. The IR field is much more complex. The heating field is more complex. And the immune response you get with all that complexity is even more complex. Now, would it be better to, or at least would it be an interesting idea to start separating out these individual mechanisms or phenomena using a much more streamlined model that is made up of 1 or 2 different cell lines, for example? Kind of going to the microfluidic direction a little bit.
So you are concerned about the different like insults, right? Like, and how they could stimulate the antigen presentation and how they can differ. There is a study done by Chi, one of the collaborators of our paper, that showed that the IRE, when you treat KPC, I think like the antigen presentation when you treat with IRE, it's more efficient when you treat with compared to cryo insults, like insults by like cryo probes, and also with thermic probes. So I think, in my opinion, if you wanted to develop mechanism of action and want to desiccate all these avenues to see like how you can tailor each component to have the best antigen presentation, I think that Chi developed a very good work that showed that IRE is providing quality enough peptides for a quality anti-tumor immune response formation. But in my opinion, we have to keep the matrix of the pancreatic cancer as complex as possible, because that's the major impediment for treatment in humans. So in order to develop a therapeutic protocol that's going to have an effective response, we need to work with all that complexity, you know, at once. Like, because my job, like my focus is usually like therapeutic response, survival, and so I want to use like tools that are being used in clinics, like the nanoknife has been used in clinics, like the IRE has been used in clinics right now to treat pancreatic cancer. And my interest is to find treatment protocols that have a high translation capacity already. So I want to work with all this complexity right now, although I know that there's a lot to be desiccated on that, but I want to see right away what are the key combination, how the drugs can be combined in a level that are going to stimulate at least some level of immune response. I understand what you're saying, that using just one cell line, and she has been done that in vitro in many of his papers, showing the quality of antigen presentation. But for me, I think I like better to represent the patient, to work with like very clinically relevant models. So for me, it's more like, how can I like overcome all this complexity, immune suppression, and like lack of drug delivery? How can my combination of treatments kind of like tilt something here, you know, that's going to work with this complexity and improve survival? So I can like, because our projects are mirror the clinics, like treatment of patients in clinics. So that's basically like why we like to work with so much complexity. You know, is that like, does it answer your question?
It sure does. So what kind of a collaboration would benefit you, since your approach is kind of different? Do you want to go top down a little bit?
Yeah, I actually really liked this tripe drug and this model of metastasis invasion, because one of the main reasons like that pancreatic cancer has a very poor successful treatment rate is because of the highly metastatic nature of pancreatic cancer. So if you can use combination of drugs and treatment approaches that would greatly decrease this metastatic potential, I think that it can be very impressive, you know. And at least for my PhD, I'm working with IL-2 right now, but I work with interferon alpha at the time. So I know the natural killer cells can have a very, very good impact in decreasing the size of tumors. So I was like, as I said to Elizabeth, maybe we could like treat animals with IRE and take that chunk of tumor and put that in a plate and then see like with different combinations how we can inhibit metastasis, you know, like for longer period of times. How long can you maintain a tumor in a system like that? Can you maintain that for like 2 weeks or so, or how is that?
Yeah, so I've carried them out like 7 days in this model. In previous models, I've gone to 14, but I would have to see how far or how long it kind of stays stable in this newer model. It's really not the tumor that I'm concerned about. It would be the blood vessel cells, like the HUVECs, that would probably not fare as well. But if I can keep a consistent like shear stress on them using a pump, it should be okay.
Okay, yeah.
I would say 2 weeks is probably like within reason, but definitely I would want to test that.
Or we can even do like something similar that she did, treat cells with different insults, like as like Alptekin said, like a IRE, thermotherapy, cryotherapy, and do like the priming of the T cells and see how well they migrate and eliminate the tumors. You know,
Yeah.
I think that would be interesting actually. You put the tumors, the cells primed with the peptides in one chamber and see how effectively they eliminate a real piece of the KPC tumor, or maybe like a human tumor, like a piece of a human tumor, right? You could do something like that, right? Or no?
Mm-hmm. Yeah, you can kind of add anything you want to microfluidic models just in general.
Yeah.
But yeah, I mean,
think that would be because he did KPC OVA kind of assays, kind of ELISA assays, you know, like the colorimetric assays. But what we can do actually is see biologically, because that's just, you see a better response and everything like that. But we could see like the biological function of the cells, if they are actually better with the different treatments, and then put them in a chamber, but then use a real piece of the human tumor, because that would be more clinically relevant, into the chamber. And then we can pre-treat these cells with these different treatment methods like IRE or cryotherapy, something that has been used in clinics, and see if there is more efficient migration and reduction of the size of the human tumor in the other chamber, right? And maybe like less infiltration of cells or prone to metastasis. I think that the only limitation would be the timeline, because it's only 14 days that you said you could do that, right?
Yeah, like that would be max. And again, that would have to be something that would need to be tested to ensure. I mean, do you need longer than that?
Well, I have no idea how much time it would take for the, like, if the tumor, how long the tumor can be alive in this method, you know, and if you would say.
The tumor can pretty much survive. And like, I see cancer cells starting to migrate and intravasate by like day 2.
Oh, okay.
So at least that's my breast cancer cells. I'm not sure, like maybe the pancreatic cells would not be as motile and therefore wouldn't spread as quickly. But yeah, it would, again, it just probably would have to something we test.
Well, this sounds like a great start. I just shared with you all on the chat, unfortunately, a tremendously long link to the application, but I'm sure if you go to the IEM website, you can find a much more reasonable, easier to digest link.
Actually,
go ahead.
Just one thing, Paulo has a collaboration with the BioNet. We can have human piece of tumors, and I've been trying to find a way to use those human pieces. So maybe we can definitely start, because now people are working, and as I said, what I believe that represents the pancreatic cancer better than anything, hypoxia, is the matrix. And this is a very good representation of the matrix. So maybe we could start doing something with the human samples and your system. And then maybe even trying the track drug, or even the track drug in combination with immunostimulatory cytokines. And then we can potentiate and kind of compare the different treatment protocols and the size of the PDAC tumor, human tumor in that chamber, and see if there is less invasion. And then we can take that tumor and stain for Ki-67 and like actually the metastasis marker like snail and stuff like that, and see if there are less metastatic markers in those human tumors. That would be actually very impactful, in my opinion. I don't know, right? I mean, something like that.
Yeah, I like that idea. Yeah, for sure.
Yeah, I kind of like that.
There are 4 or 5
like
ideas. Yeah. You guys can even get together and write an R01 on this with all the data you have.
Yeah. I kind of need that, guys, so we should do that.
Exactly.
Yeah.
This was great. Thank you all for your participation, for the excellent presentations, and the very stimulating discussions we've had. And I do have your contact info, emails. If you cannot locate one another, just let me know. I would be happy to share your email information.
Sure.
So with that, I think time has come to closely bring the session to an end. And I wish you all the very best. And please write 1 or 2 proposals and see how it goes. Worst case, you get the money, then you have to do the work. So,
but about Huda, I did not understand your project like completely. That's why I didn't include your point. Nothing against your research. Okay, cool. Thank you.
Thank you.
Thank you.